So hi, hello everyone, hello and welcome, Microbehunter here, and welcome again to another Saturday live stream, microscopy live stream. And I see that everyone is already online, or at least almost everyone. Sound is good. Thank you very much always for the feedback. And today I would like to do again a little bit of pollen analysis. Yes. And maybe if we have time towards the end of this live stream, I would like to talk about a completely different topic, and also a topic that has been requested. And this topic is about a little bit about microscope safety, but we're going to see how far we're going to get. And all of the topics that I'm not able to finish today, I'm of course able to finish next time. Well, first of all, at the beginning, during the first few minutes, I usually send a greeting all around the world. Hello from Germany. We've got here hello from Montreal, Canada, the Netherlands, the United Kingdom, from Rhode Island, the United States, Vietnam, from Belgium. Okay, sound and video is fine, very good. Last week, unfortunately, one of my cameras failed for whatever reason, and all the image was frozen. So I hope that this is not going to happen today. From the UK, and from India as well, and from France. Okay, yes, and first of all, a little bit of background information. You might be wondering a little bit of what you're able to see here. These are so-called pollen baskets from bees. These are pollen baskets from bees. Now, what they are, I'm going to explain. But I would like to say a very big thank you to a friend from Germany who sent these samples to me so that I can do a live stream. I also had a telephone call with him today in the afternoon. So we had a little chat about pollen analysis and pollen sample preparation. And I'm going to simply prepare them for you here. And we're going to see how well this actually works. So a little bit some background information first. Those pollen baskets are pollen that the bees collected. Okay, and of course bees also collect nectar to make honey, but they also collect pollen. It's also an important protein source. And here they are. I put them now under my stereo microscope. Yeah, and what I'm going to do first is I'm going to actually show you how they look like not under the stereo microscope. So I'm going to show you the desk here, and this is how they look like. Yeah, so these are look like grains, and they are from a region in Germany, from a city in Germany called Tolle. And I will talk a little bit more about that. I'm going to show you some pictures, and you see that they're in different colors. Some of them are black, white, orange, yellowish, whitish. And these are different types of pollen that the bees have collected. As a matter of fact, they're bundled together and stuck together with the nectar and also the bee's saliva. Yeah, and this has been collected by the beekeeper. Okay, and if you have, and even though I'm going to talk mostly about the pollen today, if you have any other microscopy related questions that you would like me to answer, then of course please feel free to post a question in the chat. Okay, so I'm just, they look like pebbles, and as a matter of fact, they actually do, almost sound like it as well. Yeah, like very dry. Let's put them back again under the microscope here. And I would like to show you now at the beginning, I would like to show you a couple of pictures that were sent to me. But look, maybe that's also quite nice here. If you look, then you can actually see that some of the pollen baskets, have different colors. So this actually shows that the bees did not just mix the pollen of different flower species together, but were very selective. So for example, this bee here collected pollen that was mostly dark in color. And so in other words, they're fairly homogeneous in type. Okay, so what's its use? So the bees they use these pollen as a food source. Okay, and basically they are using them as a protein source, for example, and beekeepers they collect them. Okay, so I'm going to start. I'm going to Yeah, how they harvest it. This is a question. How are these pollen harvested? And for that, I would like to show you some pictures that were sent to me. And I would like to do the following. I would like to say a big thank you. To two gentlemen here, Frank Benzenberg and Brother Markus. Thank you very much for allowing me to use the pictures. I did not contact them directly, but they were contacted over a friend in Germany, so permission was obtained to use those pictures. Thank you very much. Okay, and I would like to simply show this to you now, how they were actually collecting the pollen, and I'm going to show you a short video now. Okay, and this here are the pictures here. This is the Benedictine monastery in St. Mauritius in Germany. Did a little bit of research, and this was actually the oldest monastery in Germany. And look here in the front, this is where they set up all of the boxes for the bees. And the bees are flying in. And in the second video, I'm going to show you how they're actually collected. Yeah, so lots of bees here. It is quite common for monasteries to not only manufacture honey. Some of them are specialized in different products like wine or cheese. But this one over here evidently produces honey. You can see that the beekeepers are tending the beehives. And then at the end, look at this. Without gloves or anything, you got to be pretty brave to touch those. Yeah, so this is a very beautiful place, this is basically where bees are being kept. And I'm quite sure not only for the honey, but look, there could be some. I don't know. Did I just see some fruit trees? Maybe I don't know, apple or pear trees in the background. So it's important to have those insects, those bees, also for the pollination of fruits and of trees. Otherwise, you're not going to have a harvest. So it's not only the honey production itself that's important, but of course also for pollination. And if the bees are missing, or if there are not enough insects, then essentially it's going to be a very labor-intensive spring, because then the farmers have to pollinate the plants by hand. It means you have to take a brush and then you have to do this manually. Every flower has to be pollinated separately. Okay? So now a little bit more technical over here. This is a chamber where the pollen sacs are collected. And basically, it was explained to me like this, that the bees, they crawl through here. And I suppose that these little holes here, this is how the bees, they have to crawl through that. And as they crawl through those holes, those pollen sacs are collected, harvested. And I'm going to, that's basically how it looks like. And when they crawl through the narrow, I don't know if there should be now also some kind of a sound maybe. I don't know if it's too loud or not. Yeah, I'm going to go down with the volume a little bit here. I'm going to pause, because I would like to show you something now. Look here, this bee over here. How can I move the arrow? Look, this bee over here on the leg. Do you see this? This yellowish, that is the pollen sac. Yeah, this is how they collect the pollen, and when they enter this the pollen trap, as it's called, then this basically is removed and it can be collected. Yeah, so I suppose, and I don't know exactly, but I suppose that maybe the bees have to crawl through a very narrow opening, and then this kind of catches those pollen sacs and they kind of fall off, and then this is how they're collected. Okay, let's move on a little bit with the video over here. Okay, let's move the arrow out of the way. So the question is are they useful for us humans too? Honestly, I don't know about that so much. Okay, so but over here you see how they are actually collected. Yeah. I do have to hypothesize now, and maybe some of you who are keeping bees, maybe you can give me more information. I was not able to find any info on that. I suppose maybe if you remove those pollen sacs, those pollen baskets, maybe you have a stricter control of what the bees are able to eat. Because usually you give them kind of sugar water, so maybe that has a reason to do with that. Yeah. So are the pebble-like things pollen? This is another question by Tini, and the answer is as well, they are pollen baskets, and this means that there are many pollen packed together, and that's why I want to put them under the microscope so that you actually can see the pollen, okay? Because these are just clumps of pollen together. Right, so this is actually quite nice. And what I'm going to do, I'm going to go back to the desk here, and I'm going to just show you what I've prepared 3 slides already. I have over here a coffee warmer, which some of you might already know. This is a coffee warmer. You can also put this thing on here as well, and I'm using this because it is very, very ideal for drying slides. It really is a big help. There are those versions available that have a USB connector, but I wanted to have more power, so this one is actually directly plugged into the power source, and I'll be using that to actually dry the slides. Okay, and what I would like to do now is I'm going to simply make a couple of those, and that's the next thing, because it's not the first and also not going to be the last time that I'm going to prepare the pollen, because I would like to test this a little bit. I'm going to use this mounting medium over here, which you already also know, some of you. It's the clear glue, Elmer's clear PVA glue, but I'm also now ordering a so-called glycerol gelatin or glycerin jelly, according to Kissa. And this is the glycerin jelly, according to Kissa, is the standard mounting medium used to mount pollen. And what I heard today in my telephone call with a friend in Germany, he said he tried that, and the pollen, they look so good, no comparison, much better than if you are to mount them directly in water. Okay, so this is my little project for the upcoming weeks. Okay, So, somebody says that it can be eaten. Yeah, okay. I mean, I guess you can eat pretty much everything. And before I do that, this is basically, again, it's I got this also as a present here. And I would like to simply show you what the products that they're making here. This is actually the honey that the monastery is producing. And it's a high quality honey, so I'm quite happy that I received this as well. Yeah. So what I'm going to do also in the future is I'm going to take a little sample here, mix it with water, and centrifuge it, and I'm also able to directly extract the pollen from the honey as well. Yeah. So that's basically the thing that they're producing. Let me refocus this a little bit. Okay. Yeah. Benediktiner Abtei St. Mauritius Tolle, that is a city in Germany. Okay, so let's do the following now. Let's do a little bit of pollen preparation now. I already started I've got already 3 of them here, which are left out for drying, but I'm going to simply take fresh samples from here. And what I'm doing right now is fairly easy. I'm simply going to put a drop of water on the slide. So I do have a little jar of water here. And I'm going to do this the fairly easy way. I was told today that it's actually a good idea to kind of spread then the pollen suspension across the slide, but because of the surface tension, it didn't spread evenly. So what I'm going to do is I'm just going to let it dry like this. I take a tweezer. I'm a little bit, my eyesight is a little bit bad, so I have a problem. I'm going to take it a little bit by luck. And all you do is you put the pollen basket, and you dip it into the droplet of water. So this basically means that some of the pollen are going to come off. And I'm going to simply save this one over here by putting it over here into the Petri dish for drying, so that I'm going to be able to use it later. And what I'm going to do now is I'm not going to put a cover glass on top, okay, because I want to dry it here. So I'm going to switch this on to 40 degrees, and I'm going to just put it directly under my compound microscope, simply to see if pollen are on here in this droplet of water. Okay, so that's all I'm doing. So I'm going to go into the scope view. So, and I'm going to put it now directly, and then let's see if pollen are present, and of course they're present. Yeah, you sometimes, if you're lucky, you're also able to see some bee parts. Yeah, it's obviously being a natural product. There are also some other components in there. Apparently, there are also some occasionally some spores of fungi on here as well. Okay, and did I just see something strange here? Okay. Because occasionally, I was just wondering if I was able to see actually already the formation of a pollen tube, but it wasn't the case. Yeah, so you see that's yeah, quite a nice density of different, yeah, mostly same, but yeah, but sometimes a few pollen are different types of pollen that you're able to find here. Yeah, but I would like now to permanently mount this, and for this I would like to dry it first. And drying it also makes sure that everything is nice and flat, of course. Yeah. So why are you drying the sample on the slide? Well, because I'm going to be drying it because I want to add this mounting medium on top. And essentially, if I do not dry it, but if I keep it liquid, then I have to mix it in somehow with the mounting medium. Yeah? And then sometimes this causes air bubble formation, and this further dilutes the mounting medium. It's not a big problem, I think, because this is a water-based mounting medium. Yeah, but yeah, essentially, yeah, I'm normally drying. If you carefully dry it, then also the pollen are all very flat. Why do the pollen baskets have different colors? Well, because that's actually an interesting point. Because the bees collected them from different flowers, and the fact that the pollen baskets actually have unique colors shows that the bees did not mix the pollen. Yeah, so if they were kind of collecting them arbitrarily and mixing the pollen of different flowers, then I don't know, it would be kind of all the same color, right? But you can see that yeah, the colors are quite distinct. Yeah, I can show you again the colors are quite distinct, and this maybe you can also go like this here. Yeah, here we go, right? And this kind of shows that the bees were kind of yeah, always collecting pollen. One bee was always collecting pollen from the same type of flower. Yeah, it's I don't know why they do that. Honestly, what advantage it could have to have essentially fairly yeah, uniform pollen baskets. I don't know. Yeah. So, but in any case, I'm going to now do the following. I'm going to take it away again, and yeah. So, I mean, you know what? Just because we're at it. We can of course try also different settings of the condenser. Yeah, but you get it. Okay, so I'm going to now do the following. I'm going to place this on now the. I'm going to always have to switch back to the desk view. I'm going to simply place it now on the coffee warmer at 40 degrees. Yeah, and for a couple of minutes. But yeah, you know what? I'm going to do another one. I'm going to do 3 of them. By the way, for your info, I have already prepared 3 other slides, and they are already dry. Yeah. So, and let's do another one. And I'm going to grab the next one here. I don't know, maybe a black one. No, you know what I'm going to do? I'm going to not touch those. I'm not going to touch those. Because I want to keep them as fresh as possible. I already have a few of them over here. So this one was the last one. This one is a little bit more orange in color. Oh, this one was already so soaked that it completely disintegrated. Okay. This is gonna be a super high density going to be a super high density. No, this is going to be way too. So what I'm going to do now is the following. I'm going to simply remove some of this here because this is going to be way too much. Yeah, and I don't know. Yeah. I'm going to simply transfer to a different slide now. Okay. And this one I'm going to change the tip, and I'm going to add a little bit more water. Because this is so dense that I'm probably not able to see anything, but maybe the advantage is that maybe it's easier than to spread. Okay, because what I was I have to change the tip again because it's been contaminated with pollen. And I'm going to also add. So what I've been told is the following. What you should try to do is just like streaking out and making a blood smear. You take it, this one over here, and you kind of pull it across like this. But what I found occasionally is that because of the surface tension, it doesn't spread quite evenly. Okay, so this is a little bit of thing. So I might have to prepare the slides a little bit with something so that the surface tension is broken. Yeah. So, and I'm going to also put those here now on here. And the first one, can you believe it? The first one seems to be already finished. Yeah. So let's try it here. And I'm going to make I'm going to quickly check again just to make sure that there is something on here. Yeah, here it is. Where is it? Here we go. Okay. And now I'm going to try to make a permanent mount. Okay. I'm just also reading at the same time with one eye. I'm also reading the chat. Just Google pollen color chart. You will see some nice color examples. That's good. By the way, pollen identification is apparently a pretty advanced topic. So in other words, if you have pollen grains, and if you want to simply by microscopic observation identify from what plant they are, this is a pretty specialist topic. Because especially if you have absolutely no clue of what it could be, because many pollen, of course, look the same. And by the way, this is a similar case also with other things under the microscope. Sometimes people write me an email, they send me a picture, and they would like to know, okay, which type of bacterium is it? And I have to tell you honestly, there are so many of them out there. It is even theoretically impossible to identify them with a microscope. Some of them. So it's not even a question of having all of the information, but simply it's theoretically impossible. You can also not identify the brand of a car. It's the example I always use. You cannot identify the brand of a car based on its color. Just because it has a, blue color, you cannot say which type of car it is, right? Because color itself is not sufficiently specific. Yeah, and we have the same thing also under the microscope in many cases, that the shapes and that we're seeing are not specific enough for us to actually make an identification. So what you have to do is you have to either use some specialist staining techniques, or of course these days DNA analysis might be necessary, or some chemical analysis. Yeah? So I'm going to place a small drop of this glue on here. I diluted this glue a little bit with water. This is a standard children's glue. It's PVA, contains PVA, and it works reasonably well, actually surprisingly well. It does have a few disadvantages, namely that it does like to make some bubbles, but I would say that the overall advantages outweigh the disadvantages. Yeah, and the advantage is that it is entirely water-based and it does not shrink. So, and I'm going to now do the following. I'm simply going to drop the cover glass on top, and hope that the bubbles, the 3 bubbles that you see here, are pushed to the side. If not, it's also going to be fine. Okay. The mounting medium is not only important for protecting the specimen, but it is also a question of refractive index. So usually it also improves then the image quality of what you're able to see. Yeah. So with that, on now, let's have another look under the microscope. And here we go again. Yeah, and you see that the pollen looks much brighter. That's very typical. Usually, if the difference in refractive index is too high. So, for example, if a specimen is in water, then it looks, or in a water-based mounting medium, then it looks much more clear. So one disadvantage that some mounting media have, and that's also one of the reasons why I'm testing this, is that of course it might change the shape of some of the pollen, especially when it starts to dehydrate them. Yeah, but I think over here you can see that it's fairly okay. Yeah. Can the heat damage the pollen? If the heat is too high, well, yes. That's why I've essentially done the follow. Well, let's put it this way: too high heat will denature proteins, and if the heat is too high, then indeed it might cause the pollen to lose maybe too much water, and then it might shrink too much. But that's why I left it here at 40 degrees centigrade, which is kind of a careful accelerated drying. And as long as the water droplet is wet and contains water, usually a lot of the heat is also used for evaporating the water, so the temperature does not go up too much. Yeah, it's cold. Yeah, but once it's dry, like over here, I'm going to simply take it off. This one is very, 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 very dense. Okay, I don't know if maybe it's too much. I don't know. Yeah. So I've got 5 slides now over here, and this is basically the first one. But you know what? I'm just going to leave it here, and I'm going to have another look. Yeah. See, that's a bubble here. Yeah. And then when there are a whole bunch of bubbles. And then with glycerin gelatin, I'd like to see then how it looks with that as well. Yeah. I'm quickly reading through the comments. Further up here, sehr interessant, very interesting, but where do the pollen baskets come from? The flower? Well, no. Ultimately the pollen comes from the flower, but the bees collect them. Did I not? Oh, I think I forgot to show you. Look. There's this picture I found online. Look, here it is. Okay, that's a Creative Commons picture. Okay, that's of course the bee. And look, that's the leg here, and this is where the bee has packed the pollen. Yeah, that's one of those pollen baskets or pollen sacs. Okay, that's basically the pollen basket, which basically sticks to the leg of the bee, and that's how it carries it home. I've seen some pictures there; they can be huge. Some of them. Surprising that the bee is still able to fly. So, can you stain the pollen? Yes, you can. As a matter of fact, I do have a stain. Maybe I'm going to try that as well. Just a second. As always, I'm super well prepared with everything in my cupboard. That is methylene blue. I can also try that, of course. Okay, but not after I've made already the permanent mount. So that's basically how it looks like. Okay, and it looks okay. And then usually after a couple of days, we have to check it again to see if it actually works. I'm not going to put it on this one. I'm not going to dry the glue on the coffee heater now. Okay, because I don't need that speed of drying. So we're now going to try the following. Yeah, I'm going to put now this here, which I also made before under the microscope. And then maybe I'm just going to stain one of them as well, just for the interest. Okay. So this is directly, here we go. Okay. This is how it looks like. Okay. So there's a fiber on here. I think this could be a contaminant on the microscope slide. And notice, of course, that the pollen looks relatively dark compared to the background. And that is because of the difference of refractive index. But it looks better if it is submerged in a mounting medium or in water. And as I just mentioned before, glycerin jelly, according to Kissa, that is the standard mounting medium used for pollen. Look at this. There are some of them are much huger, much larger than the others in here. Yeah? But you can see that actually the pollen itself is mostly of the same type. Yeah? So let's try it again. Okay. So I always have to switch back and forth. I would like to have an easy way of actually switching the scenes here. You know what? Let's try the following. I'm just going to try. Should I try? I'm just going to try to stain it. Okay. Again, I've not tried this yet, but what I'm going to do is I'm going to put a little drop of this Löffler's solution on top here. It does contain alcohol. So this is always an issue with stains, that stains might actually introduce so-called artifacts, which means that they might deform the specimen. They might not only stain them, but also deform them somehow. The alcohol might withdraw some of the water. Sometimes the alcohol that's included will, in the stain, might break open the cells. So it really depends quite a bit. And what I do now is I will simply take a tissue paper after a couple of seconds, and then you should dry it again, and then you can actually put some mounting medium again on top. So I'm going to simply do this the very easy way. I'm just going to pour it off. Maybe it would be a good idea to give it a little wash. Okay. So I'm going to add a drop of water here. This is of course very risky because you might wash off the pollen. This has not been heat fixed. In the case of in bacteriology, in microbiology, what you do is you sometimes heat fix the cells. This basically means that you pull the dry slide with the specimen through a flame, and this bakes the cells to the glass slide so that you can stain them without them being washed off. So there's a tiny droplet here on the side. I'm going to just put it again under the microscope just as it is. Then I'm going to dry it, and I'm going to make also a permanent mount. So here we go again. Yeah, and that's how it looks like. Yep, they are stained. There is now water on here as well. I have to be a bit careful that I don't touch the... Yes, here we go. Yeah. Yeah, because this is very risky, because I'm now using the 20 times objective, so the distance is very low, no cover glass. Okay, so this is how it looks like, and it's fairly stained fairly darkly. The idea of staining, of course, is that the specimen looks then darker stained than the surrounding background. So, and before I make a mount here, I'm going to put it also again on my coffee warmer here on top. You can see it. So that I'm going to carefully dry it, and then I'm going to again make a mount. So this one over here is one of the things that I just made recently, which is extremely dark, extremely dense rather. No, but it's okay. It's okay. Looks worse. It looks worse under, looks worse when I did not put it under the microscope. It looks very dense, but actually it seems to be okay. So let's give it another try. And that's the wrong one. This is the methylene blue. I wanted to actually use the mounting medium. Okay. So I'm just reading again they form, okay, yeah, okay, there seem to be no new questions here. I do receive emails, I receive comments, and some people, have the problem that the mounting media and that I sometimes use, like for example, when making mounts of insects, I really like to use the mounting medium Euperol. But there is sometimes the problem that some of these substances are very difficult to obtain. They are available. It's not that they're rare, because they are commonly used, but sometimes companies will not sell them to private individuals. They only sell them to institutions, right? And so this can indeed be a challenge, and for this reason some people essentially send me emails and says, yeah, but you always talk about those mounting media, but the problem is we cannot get them. And this is indeed a problem. Okay, and for this reason, I do recommend to use alternatives. And this one is one of those alternatives where you can buy a bottle on Amazon of Elmer's PVA glue, clear glue, not the white one, but the clear one, for really a very cheap price. So, and this one is also it's a little bit not straight, the cover glass. So let's And I do have to label them as well. So but let's put it again under the microscope. And here we go. And again, you do notice that they do look somewhat more transparent and brighter. Okay. Yeah, there seem to be different kinds in here as well. And sometimes it's also good to do a before and after check, because some of them will actually change their shape. Usually it's like this, that the pollen, when they start to dehydrate, some of them will start to shrink, and if you add again water, they will start to swell. So they do change some of them do change their shape as well. Maybe this is one of the reasons why identification based on shape might be difficult. Yeah? So basically the pollen is moistened by the bee's saliva and is stuck to the hind legs. Yes, that's how they create pollen baskets. Yes. What I heard is the bee's saliva and also nectar that they collected. That kind of forms some kind of a glue. And this is how they make those pollen baskets. Yeah. So this is what I've also done before is I once used I think also Uperal mounting medium, which does contain an organic solvent to mount those pollen. And then what happens then is that sometimes if the mounting medium contains a solvent, then the pigments that might be present in the pollen sometimes also start to leach out and start to become dissolved. Yeah. So you have to try out what's best. But again, I know I'm repeating myself. The standard mounting medium for pollen is glycerol jelly, which, however, unfortunately, it is a little bit difficult to use because you have to warm it up to melt it. Yeah? So that's basically the other one over here. The one with the stain is finished. So this is slide number 2. Slide number 3 is the one that I stained. And where is the glue again? And I'm going to put that on. Now, I'm going to make now a mount of this one here. It's a little too much, maybe. Maybe a bit too much. Yep, let capillary action do its job. It now spreads, and but I think it's okay. And we go again under the microscope. And here we go. So that is the stained sample. And I don't know, it does look quite dark. It does look quite dark. Yeah, I mean staining. I mean that's the whole point of staining, right? You want to make you want to increase the contrast, but maybe in this case it's a little bit too much. I don't know. I'm going to now open this here up. I'm going to even now remove the polarization, so that's now bright field. So now it's much better possible to see the actual blue color. Can we get rid of the air bubbles in a homemade vacuum chamber? Yes, yes, kind of, and no. Generally, you should always, if you make your own mounting medium, then you should degas it in a vacuum chamber. But this will not always help with bubbles that are introduced when you make the slide. It's definitely better than nothing, because if the mounting medium is degassed and does not contain gas, then it might be that maybe some of the bubbles that are in there might also start to disappear. Yeah, you can see that in this case, some air bubbles are caught between the pollen. So this is a little bit somewhat of an issue. It's not a big problem, I think, because you're always going to find places, that don't have air bubbles, but it kind of shows a little bit that mounting medium and the specimen should be a little bit compatible and mix. Yeah? It should be possible to melt the glycerol jelly directly on the slide on the coffee warmer at 40 degrees. Yes, this is actually what I would like to try. You can either take a small amount of solid glycerin jelly and directly melt it on the slide, or use a warm water bath. However, another thing is, some of you might know that I'm a teacher, and we've got a lot of equipment and substances and mounting media also in school. And I found a bottle of glycerol jelly in school, way overdue. I don't know, it's all the way in the back of the cupboard. And unfortunately, it did not turn solid anymore. And I think what might have happened is if you overheat gelatin, then it might not solidify. You're breaking it, yeah. In other words, and it might not solidify anymore. Yeah? So this is also an issue with this type of mounting medium. You have to be careful that you don't heat it too much, because otherwise it might not work in the future at all. Yeah? So I've to be honest, I've never tried to work with glycerin jelly before. But it is kind of on my list of things that I would like to try out. And now with so many pollen here to mount, that's maybe going to be the contents of another live stream, maybe in a couple of weeks. Okay, so, but glycerin jelly, just for your information, why some companies don't like to send them, because glycerol jelly is a water-based mounting medium. Therefore, it means that theoretically it can turn bad, it can spoil. And fungal growth, for example. And for this reason, in the original recipe of glycerin jelly, they added a substance called phenol, which is not healthy when you inhale it. Yeah? And for this reason, some chemical companies do not like to sell it. But apparently more modern formulations of glycerol jelly do not contain this anymore, but contain safe alternatives. I thought glycerol jelly was always a liquid because it always absorbs water from the environment. Good point. Glycerol jelly is a semi-solid. It will turn solid, but it will absorb water as well because of the glycerol. So when you what you do is you have glycerin jelly, and it is solid because it's cold. Then you warm it up. You make your microscope slide, and then it will turn hard again. And a little bit of the water will evaporate, but it will not turn completely dry, because it will absorb, as you correctly said, it will absorb water from the air, but it will not be completely liquid. Yeah, so that is the thing. Yeah. With which samples can we use? In eosin, eosin and hematoxylin and so on are also standard stains that are used for differential staining. They're also a very general purpose. I think eosin, I've never used it before. It can be used, I've heard people using it for tissue staining, also plant tissues. It is certainly something that I'd have to research this a little bit, what substance it actually stains, what chemicals it stains, because the different stains, they react with different substances inside a cell. So for example, the methylene blue solution that you see over here contains alcohol, and it contains also potassium hydroxide, KOH, so it's alkaline. And it will therefore react very well with acidic parts of the cell. And this, for example, would be DNA. DNA is deoxyribonucleic acid. It's an acid, and therefore methylene blue in this case is known also to stain DNA. So you have to basically choose your stain based on that what you actually want the stain to react with in a cell. Okay, so that's a little bit that's a little bit the thing, and So eosin, hematoxylin, methylene blue, general purpose stains, and usually if you try them, then you can actually see sometimes with hematoxylin and eosin a red-blue combination. Yeah. So let's do the following. Yeah, that's basically now methylene blue stained, right? And I think we get the idea. The pollen now looks blue. So, and I mean, I do have 3 more slides here. I'm just checking the time as always. Okay, 44 minutes. So maybe for another 15 or 20 minutes, I'm going to make a few more, slides here. And then I am going to make a very large jump in topic. And I'm going to move over into the theory part. Because I do want to talk a little bit about my opinions about growing microorganisms and safety issues, as it relates to microscopy. So I think it's quite obvious that there are no major safety concerns here with what I'm doing here, but with some microorganisms you've got to be a bit careful. And I'm not saying that it's always extremely dangerous, but I would simply, because it's just general hygienics, to avoid possible problems. Yeah. I mean, I have to, being a teacher for a school, essentially, no, I lost my cover glasses. Being a teacher for a school, of course, safety is absolutely important. And for this reason, for example, there are certain mounting media and so on that we simply don't use in school. Yeah? And because it's simply not necessary. Yeah? So, and that's also was one of the reasons why I was kind of searching around for safe mounting media and safe stains that really are not a problem at all, even if children completely use it improperly. So I think I'm just going to leave it at that now. Let the mounting medium spread. I'm really happy. I mean, I know I'm repeating myself, but this PVA glue is really a lifesaver. Not lifesaver, I'm exaggerating. There are not many water-based mounting media out there. And and this one actually is, because this basically means that the specimen does not have to be completely dry. Yeah? Otherwise, there is a preparation steps necessary as well. Yeah? So, dehydration steps, that is. So here we go again. So this is way too bright. So, okay. Yeah, see, that is... Occasionally, if you are lucky, you are able to see some hair from bees. Oh, look at this. This here is clearly shows that the mounting medium was not able to reach all of the pollen. Lots of air bubbles in between, probably also because of the density of the pollen. Yeah, so they're all air bubbles in between here. Yeah? It's not a big problem really. Yeah. Why not suspend the stain in water instead of alcohol? This is also possible. But Löffler, this is methylene blue based on according to Löffler, he devised a stain that contains alcohol and potassium hydroxide, and I assume, I'm not guessing, okay? I assume that maybe the addition of the alcohol allows the stain to go into the cells better, because the alcohol dissolves away a little bit the cell membrane. My guess. Yeah, but it is, of course, methylene blue can of course also be in water. Yeah, see this here, a very nice example of way too many bubbles. But we have to see a little bit how this actually develops as it dries. It could also be, and that's also important, that not all pollen like to be in contact with water all the time. So it depends also on how hydrophobic they are. If the specimen is very hydrophobic, then it does not mix well with a water-based mounting medium. The extreme case that I've once shown is of bird feathers, for example. They're greasy, they're oily. If you use a water-based mounting medium, they really start to stick together, and there are lots of bubbles. Yeah. So let's go up a little bit further so it's now 40 times, and yeah. Here, this is how it looks like. Yeah. Let me check again. This is now bright field. This is basically how most of you probably are going to see those pollen, because most of the people have just regular bright field microscopes. Unfortunately, I'm not able to add a Reinberg filter to this particular microscope, but by using color filters, you can of course also change the colors around. Yeah. Do pollen have half part DNA just like sperm? Ooh, that's an interesting one. It's more complex than that. So the question is for fertilization, sperm and egg cells only have half of the DNA, they're haploid. But actually what happens with pollen is that a pollen tube will grow, and in the pollen tube there are two sperm cells. Two sperm cells will form. One will, and they're responsible for, one of them fertilizes the egg cell, the other one fertilizes the so-called the endosperm of the ovule. So this basically means it's more complex than that. The pollen itself is not yet the gamete, but it will form the sperm cells. Yeah? So this is a little bit more complex. It's been a long time ever since I studied botany at university. But I had to know all of those steps as well. And while it is correct that pollen is, of course, the male part, it is not equivalent to a sperm cell, but it will actually form two sperm cells. Yeah. Does it make sense to support the hardening of a slide with PVA glue on the coffee warmer to support the? So that's essentially the question. I didn't try that, but what could be is, and I don't know if this is for all practical purposes possible. Could be even an advantage if you start to warm it. What happens to the air bubbles? They might also expand a little bit. Okay, during the heat, maybe this allows them to escape, but then they might collapse. Yeah, so this is indeed something I did not try yet, but we could try it. Yeah, I'm just going to put. I'm just going to take this one out and maybe put it on the coffee warmer. But sometimes that's so I put it. Sometimes it's maybe also a good idea. What I read once, not to dry them too quickly, because you want to give the mounting medium and whatever solvent it contains enough time to actually also go into the specimen. Yeah. So this is But then again, having said that, there are mounting media out there that are specifically designed for very fast drying, because you need them for routine observations in a hospital, for example, and it's not necessary for the slides to store 100 years, but just for a couple of weeks maybe. Yeah? Can you find DNA in fossil pollen? And the thing that's now my view of the issue, if the structure is sufficiently preserved, if the original structure of the pollen, not just an imprint, but if the original material of the pollen is sufficiently preserved, then actually also DNA should be present. But what I also heard during my studies of molecular biology is that the problem is that the DNA can be quite fragmented, because it will start to decompose and fall apart. But they've even extracted, they have extracted also DNA and even protein sequences from very old fossilized bones as well. But my personal opinion is you got to be really careful extracting DNA from fossils, because there is, of course, always a risk of contamination. So then you don't know, are you really extracting the DNA of the pollen or some kind of a contaminant? Yeah, so that might also be an issue, and this possibility also has to be excluded. Yeah. So Edith Haviland is posting a question mark. Did I oversee something or not? I don't know. In those triangle pollen, can we see some holes on the corner? I don't know. I don't know where it is now. I'm just going to put it back again. Yeah. So, and let's have a look at the scope. So let's go up with the magnification. Yeah. Of course, the interesting thing is now if I were to put those into a sugar solution, those pollen from the bees, am I then able to trigger the formation of a pollen tube? I don't know. Yeah. Or could it be that specifically maybe there is something in the saliva of the bee? Again, I'm totally speculating, which prevents the formation of a pollen tube. Yeah. Can you please tell us why they are triangular? Okay, this is getting philosophical here. Okay, if let's put it this way, the question is now why are these pollen triangular? And I would like to respond now, and I'm not trying to be provocative, but I am responding now. Why not? Yeah, why not? If they had a different shape. Then, and if they would do their job, then essentially we would ask why are they round, right? So essentially what I'm trying to reformulate now your question is, if you're asking a biologist why is a certain structure the way it is, why are these pollen triangular? If you were to translate this into the language of a biologist, you would say, what is the evolutionary advantage of this pollen to be triangular? This would be kind of the language of a biologist. What's the evolutionary advantage? What's the selective advantage? And sometimes there is no advantage. It simply works the way it works. It's a little bit like asking, and because this is a very common question that I'm being asked, what's a certain advantage? It's basically almost like, and I always ask back, what is that? Why do we have two eyes? Why don't we have a third eye up here? Then wouldn't we be able to see better with eyes in the back? And the answer is it works the way it works right now, and nature is simply very diverse. Yeah? So this means sometimes there is no specific advantage. Yeah? Sometimes there are simply several solutions to the same problem, and if it is not a disadvantage, then it is simply maintained. I know this might not sound very satisfactory, but the answer essentially is it's just the way it is. There might be an evolutionary advantage. There might be an advantage why it's triangular. Maybe it has to do something with the stability of the pollen, with, I don't know, yeah, maybe larger surface area allows it to be carried better by the wind. Yeah, so you can always find certain advantages why it's triangular, but then immediately somebody else is going to ask, yeah, but there are all the pollen out there that don't have that, and they also work. Right, so this is a little bit of a problem because it doesn't matter how you twist it and turn it. You can always find another way to look at the situation, right? So this is a little bit the issue, and at least when I was back at the university. I remember this actually was a course, introductory course at university, and the professor said something very interesting. He says, yeah, many people always want to look something, they see a certain structure and they always immediately ask, what's the advantage? Why does it look like this? And the answer sometimes is there is no advantage. It's just the way it is. It works, and there is no reason. And sometimes this is not a very satisfying answer in the sciences, where we think that everything has to have a reason, right? But sometimes in nature, it works the way it works. And it's an interesting topic that I like to talk about, because it's a common question that also my students ask me. Yeah, but I don't know if the answer is satisfactory. Yeah. I'm still working on fluorescent microscopy. Just in the afternoon today, I basically talked to someone who is also experimenting around with that. I've been told that by shining UV light on this pollen, this looks apparently fantastic. I have not tried this yet, and I'm trying to find a system which does not hurt my eyes. I have to be honest. If you do any do-it-yourself fluorescent work, you got to be really careful, because as soon as you're starting to work with ultraviolet light, you might actually, if it's not done properly, you might damage your eyes, right? So this is the thing. Yeah. No, you said Edith is saying, I'm sorry. No, please, no reason to be sorry. It's an interesting question. Seriously, that's why I'm getting so excited about this, because I'm fascinated by these questions myself. Yeah. Can pollen be used to learn more about climate change? Oh, not that I'm an expert in that, but I'm quite sure that if you, for example, analyze maybe the distribution of pollen somewhere, that you're able to also quantify maybe the change in flowers and in plants that are actually in a certain area. I want to tell you a story, actually, and I know I'm deviating now a little bit from microscopy. A couple of years ago, I visited, we did a holiday on a farmhouse here in Austria, where I'm living, like in many other countries. Some farmers are actually renting out apartments, and they're still running a farm to have their animals, but at the same time, as an extra source of revenue, they're renting out summer apartments. And I had the possibility to talk to that lady there and to the farmer. And she said that over the last 30 to 40 years, essentially she said is people don't notice that. They think, oh, everything's fine, everything's green. But what they don't notice is that the plants have completely changed over the last 30 years because of climate change. Yeah, so this is sometimes the cows cannot even find the plants that they're used to because of change in climate and so on. There has been such a shift in the plants that are actually growing in a certain area. Yeah? So this is, and I'm quite sure that there might be some studies out there where people are actually analyzing the whole thing over pollen. And how this can actually, the type of plants can, or the change in the type of plants can be actually monitored. I talked to an entomologist a couple of years ago, and he basically is doing a similar study with a change in insects. So he every time when he's catching insects, he's entering them into a database. And I asked him, what's the higher scientific purpose of that, of documenting this? And he says, yeah, this way you can actually trace and track the change in insects due to different climate conditions. Yeah. So this is, this is anyway interesting things here, right? But I'm not into that. How do you say detailed? Level, so to say, into that detailed analysis. I'm doing microscopy mostly for fun, and simply because I like to observe. I like to observe nature, and I like the surprises sometimes, the things that you see. Yeah, lots of bubbles here as well. Yeah. So that's for me. That's kind of a little bit the motivation here. That microscopy and nature observation kind of expands a little bit the horizons. Yeah. So what am I doing now? It's 1 hour now. So what I would like to do, I promised to talk a little bit also about microscopy safety. Last slide here, and I'm going to have a look at those slides, of course, next time again. So this is not going to be the last time. Look, this is way too dense. Big air bubble. Yeah. So I have to find yet a good way of actually spreading those pollen. They look very they look very triangular, some of them here. Yeah. So okay, big change in topic. Okay, for about 15 minutes or so, if you don't mind. And so this is It's also not only because it has been a request, but also because I do receive occasionally some emails. And it's a little bit the question now is, yes, and some people are really, I don't know, overly anxious and worried. And then sometimes I see some people on YouTube growing bacteria in petri dishes, and I say, please don't. Damn. Yeah, so I would like to simply show you the letter that I got. And I'm going to also make a shout out. Maybe you know the YouTube channel Thai Microcosmos. Okay, so here it's kind of a shout out. Yeah, it's a beautiful microscopy channel as well. Okay, so thaimicrocosmos.com. Please do visit it. Right, and this is a long email with a suggestion on what I could talk about concerning microscope safety. Okay, microscopy safety. I'd like to suggest a full stream about safety during microscopy. Now, this is not going to be a full stream, but I just want to get started a little bit, and then maybe I'm going to dedicate another stream to this, but because it's more theoretical and less observation, for this reason I did not want to spend a whole stream on this. You have a lot of viewers who are new or young or even neither, like me, who don't always take proper precautions with respect to cleanliness and safety. You often touch upon the subject in your streams, but only in passing. In fact, in your latest stream, which was last week, you gave more detail than usual about bacterial safety. I'm suggesting you go deeper, okay? So here are a couple of things about collecting specimens, how to properly store them, cleanliness during observing, after observing, how do you store samples over long term? Also equipment safety. So for example, when you should never ever touch halogen light bulbs with your bare fingers because they might explode afterwards, or not explode, but at least they might break because the oil of your fingers, when it's deposited on the halogen bulbs, will actually cause a local overheating, even to the extent that the glass can melt. Yeah, so about the whole issue about crystals. So basically, especially if you're using chemicals and you're mixing chemicals, then there might be some harmful reactions happening. Yeah, and so there are plenty of things to talk about. Thank you very much. I will pick up on that, but there is one thing that I really would like to talk about specifically, because I am doing that myself. Just a second. But I have occasionally some mixed feelings as well, okay? So what I have over here is some pond water. And what I've done is I added a crushed wheat grain. How does this look like? So it's kind of cereal grain. The powder that you see here, that is dry yeast. And all you do is the following. You take this, and I don't have spoons now here, but between two spoons, you kind of crush it, and you drop it into some pond water, and then you wait for a couple of days, right? And if you're lucky, you're going to see a lot of ciliates, paramecia. They're generally harmless, right? But why do they grow? They grow because by adding some food, some starch in this case, you're increasing the number of bacteria in the sample, and then the paramecia, which might be present, feed on the bacteria. I also had limited luck once. No paramecia grew, and the reason is because I think I had a couple of water fleas or water crustaceans in there that immediately ate them away as well. But the real concern is that if you add some organic material, some food to a water sample, and when you have an exponential growth of microorganisms, you don't know what you are growing. That is the problem. Yeah, so that's problem number one. And problem number two is that the concentration of microbes, so bacteria, can be extremely high, very high, much higher than that you would actually find in a normal pond, right? Because some people, when they say, that's what you're able to find in pond water, say, oh my gosh, so many critters inside the pond, and says, no, yeah, of course there are, but no, what you've got here is much higher. Okay, and so that is a little bit of an issue. And what I'm a little bit concerned is if I were to get it on my finger, I wouldn't worry so much about it because the skin is a very good protective layer. But what happens if there's a tiny wound, for example? Okay, or if you rub it in your eyes, you don't know what you have here. And during my while I was studying microbiology at the university, I basically was working with unidentified bacteria. It was my job to identify bacteria using a series of chemical and DNA tests. And I was growing bacteria in the lab. And I was already in the safety category, biohazard category number 2. So it was actually an authorized lab. It was because it was unknown. These were unknown bacteria. If you do regular genetic engineering, then you're using E. coli bacteria, which are well defined, and you know that they're harmless, and it's used for genetic engineering. But if you're growing wild bacteria, you don't know what you're growing. Okay, so and I do not like it when people actually grow, for example, take fingerprints, right, and put the fingerprints on Petri dishes, for example, because these are then bacteria not only growing in water, but actually are able to grow on your body, right? So generally, what you have to do is if you do stuff like this, be careful, make sure that you observe general hygienics concerning this. And I'm going to do the following. I mean, I know what I'm kind of doing because I've studied this stuff, right? And by the way, the same thing, you don't want to spoil food, right? That's not good either. It's the same thing, right? What you do is now the following. You go in here, you take a sample out, you put it on here, and then basically the tip here is completely covered in bacteria, right? And this is now an issue because I have to, ideally, I have to now, disinfect it or so, but I do have a special container. When you now put a cover glass on top, and I mean the sample is so old, so maybe it's already gone stationary, so I'm not even sure if there's so many left in here. But if you put a cover glass on top, and then when I observe it under the microscope, what am I supposed to do with this slide then, right? No reason to get paranoid, but it's like having spoiled food, somewhere, and I don't want to spread those, right? Because, for safety reasons, I'm just going to put it under the microscope now again. Let's have a look what we're able to see. Okay, and not very much. We go up with the magnification. We go up with the magnification. We go up. So that is now 40 times DIC. And you know what? I'm going to switch over to phase contrast. Back, 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 because phase contrast is very good for observing bacteria. Yeah, those things that move around might be also smaller ciliates, but look at those tiny lines here that you see. These are all bacteria. So, and which ones are they? I have no idea. Yeah, I have no idea. I'm not automatically assuming that they're a problem, but I don't know what they are. Yeah? And I've actually also seen some even spiral-shaped bacteria, spirilla. You do not want to get those on your body. Yeah? So this is a little bit, an issue. Yeah? Where you just have to be very careful. And there's no point to be paranoid. But sometimes when you go online, they're suggesting, you can do something called a hay infusion. You take dry grass, you add water to it, and then you let it stand. And then you have microorganisms growing in there. But the amount of bacteria that you're growing there is really high, and you do not know which ones you're growing. Is 99% isopropyl alcohol strong enough to clean my material? And you will be surprised. You should not be using 99% or 100% alcohol. You should be using 70% ethanol because it disinfects better than higher concentrations. This might sound strange, but the standard solution for disinfecting is 70% ethyl alcohol. Why that? Why not isopropyl alcohol? Because first of all, isopropyl alcohol does not mix so well with water. You can just use regular ethanol because the alcohol, 70% alcohol, will dissolve the membrane best. The cell membrane of the bacteria, and therefore it will actually work better in disinfecting than higher concentrations. Okay, so this is a little bit the thing here. So 70 is beautiful. What's going on here, people? Look, isn't this great? A little bit of biology again. Look at those. Okay, what do we see here? We see a whole bunch of bacteria floating by. We see something green in here. We know it's green. This basically means chlorophyll. This means photosynthesis. This means oxygen is being produced. Oxygen is being produced. And those tiny flagellates or ciliates, most likely, are now going to go there where the oxygen concentration is higher. And of course here around this algae, yeah, there's plenty of oxygen. This is the reason why they gather around here. And the reason is because this sample that I've got here is low in oxygen. How do I know that? Because all of those bacteria here consume so much oxygen that those ciliates over here have a problem. Yeah. So that is the thing here. So you can see this quite often. Yeah. That yeah. And this is traditional, this old, I forgot the name who did that, but this one scientist, what he's done is the following, and I know I'm deviating again, I'm sidetracking. He was actually shining different colors of light on those algae and was observing how many of those paramecia in this case, or in this case other ciliates, gathered around it. And so he was therefore able to discover which colors of light are most used for photosynthesis. Yeah. So basically, yeah, he discovered that blue and red light will cause most of the other microorganisms to gather around, which shows that mostly blue and red light is used for photosynthesis for the production of oxygen. Yeah, just some side issue. Yeah. So theoretically, it's not completely safe to save and examine water samples for a long time that you occasionally feed with crushed grains over the years. It depends. Look, this is that's why it's so complex. This sample that I have over here right now on my table contains a lot of bacteria. But if your water sample contains a lot of ciliates, is rich in oxygen, then those ciliates or water fleas, water crustaceans will keep the bacterial concentration low. Yeah, because the bacteria that will divide. So it is not a question of age. Because even old samples can have a low bacterial count provided that the whole system is in balance and provided that there is a so-called a top-level predator like a water flea, water crustaceans, or ciliates maybe that eat the bacteria. So and in order to promote the growth of those larger organisms, you must make sure that there is plenty of oxygen in the water. So it is not a question of age. I've got a whole bunch of water samples over here that are, I don't know, months old, but the bacterial count is fairly low. Look at an aquarium, for example. Many people have an aquarium at home, right? And it's perfectly fine. But the problem is if the bacterial concentration due to overfeeding reaches a certain critical point, then the system might tip over. All of a sudden, the oxygen concentration will go down. All of those higher organisms that eat the bacteria will die off because they need a lot of oxygen. Bacterial population will explode, and then basically that's what you call, in nature, you call eutrophication, is the death of a pond. Right? It's basically plenty of bacterial growth. Yeah. So this is the thing here. Yeah. Why does it look like that the microorganisms are flowing upwards? Yes, this is simply because of capillary action. If you look here at the top, right, sometimes it takes a little bit of time for the water to spread evenly and or of evaporation. So this means if it starts to evaporate on one side, then this also causes a water flow. Sometimes after a couple of minutes, we might actually see that this whole system stabilizes. And look, there are more and more of these now actually collecting here. I'm going to go, yeah, okay. There are more and more of these here now collecting here, yeah, because, yeah, definitely because of oxygen production, yeah. This morning I squashed a diatom under cover slip, so I've seen the two parts of the glass. Ah, okay. Yeah, I was excited as I'm looking, yeah. So basically the thing is that's nice. Diatoms actually have those diatom shells that kind of fit together like cardboard boxes, right? And here apparently you applied too much pressure, and then this kind of you were able to separate the two halves of the diatoms. Yeah, yeah. So yeah. Again, I've got water samples here that I've just also made I don't know, this one water sample with a water fleece. I don't think it's 1.5 years old already. I'm feeding it all the time. But the bacterial concentration is fairly low. There are lots of algae in there, but the water fleas, the ostracods in this case, they basically not only eat the food that I add, but they will eat also the bacteria. Yeah. So you see, it's a question of keeping everything in balance. They're green. This basically means lots of photosynthesis, lots of oxygen, and then they're happy, and then they will keep the bacterial concentration low. Yeah. But as soon as you start overfeeding, then it might actually tip over. Yeah. So this is a little bit, I know I didn't go into very much detail concerning feeding it. Yes, I'm feeding my water fleas, yeah, because I want them to reproduce. Yeah, so maybe not always necessary to feed them, because if there are plants in there, algae in there that actually are primary producers, then they will feed on the algae. But I decided I want to have more. I add a little bit of yeast to the system, and then what they will do is they will actually eat that. Yeah, yeah. So there are multiple ways how you can approach that. But yeah, again, just wanted to demonstrate here the large number of bacteria that we have here, which are those tiny string-like structures here. Okay. Could you show us some of your samples? And yeah, I'll look at this. Do you see this rapid streaming? Yeah, this sometimes happens if an air bubble somehow pops somewhere, or two air bubbles fuse together, and this causes sometimes for a short burst of rapid streaming. Yeah. So sometimes water evaporates and there are air bubbles and they kind of collapse, and then you have a very rapid streaming of yeah. So let me quickly go back here. Yeah, I will, yeah, not a safety issue, but I read that you can get motion sickness from looking into a microscope, especially when the stage is moving fast. I've luckily not experienced this, but it doesn't surprise me. Once, many years ago, I watched in a cinema, I watched a movie, and the camera movement was very rapid. And I actually felt a little bit sick. I had to close my eyes. Yeah, so it doesn't surprise me. Because I've experienced something similar, maybe not so much with a microscope, but actually, yeah, because what happens in this case, people feel motion sickness because the brain gets conflicting information. The eye sees a movement, but the balance organs in your ears, they do not sense this movement, and therefore this is a conflicting information for the brain, and then people can start to feel sick. This is, by the way, also motion sickness on a boat, for example. The boat is moving, the sense of balance in your ears tells your brain the boat is moving, but the eyes actually give you a steady view because you're moving with the boat, so everything is steady, and this conflicting information causes some people to feel sick. I've never heard that this happens with people using a microscope, but it doesn't surprise me. Okay, it actually makes sense. If you're moving around all the time with a microscope and then you see a vision, yeah, but the sense of balance tells you actually you're quietly sitting on this chair, then of course I can imagine that some people feel sick because of that. I felt sick at the movies once. Yeah? I had to close my eyes. Yeah, so yeah. So people, what I'm gonna, yeah. I'm going to call it quits today. And again, I only touched the safety aspect a little bit. I actually wanted to talk a little bit more. Yeah, what I'll do is I think I'll pick up on that a little bit more later, because I do consider it an important topic as well. But my suggestion is another thing, maybe as a final thing, is how do you dispose of this now? Right? If you really want to get rid of, and you got to be careful still, if you really want to get rid of bacteria in a very efficient way, also if you've got somewhere in your house, maybe in your bathroom, some mold growing on the wall, a very efficient way is not only alcohol, but is hydrogen peroxide. But you got to be so careful with that stuff. Really, it's really aggressive. If you get it in your eyes, it's a real problem, right? But hydrogen peroxide is indeed very effective in removing, microorganisms. Yeah, but yeah, it's the question again is if it's worth it. You can use also some other relatively aggressive substances containing chlorine, but that is environmentally not always the very best thing to do. Yeah. So yeah, I'm just saying. I think what I will be doing now is I'm going to wait for the slides to dry. We're going to check again next week. I might pick up a little bit more on this whole safety aspect. Okay. But I still wanted to talk a little bit about that because when I show things like enrichment cultures, we're adding some food. And people are copying it. You got to be, healthy respect, but not become too worried either, right? So what I'm going to do is, yeah, I'm going to call it quits for this week. I hope to see you again next time. I'm glad that the stream worked out technically and that no cameras froze. Okay, if it is necessary for me to change the date of the live stream, then please do check the community posts or the announcements. But for today, I'm going to call it quits. Happy microbe hunting as always. Do also watch my other videos on this channel. Okay, see you around next time. Bye-bye.